So the next organelle is the ribosome. So ribosomes are nothing but granular structure. Okay, and this also was first time observed under the electron microscope. As a dense particle. As a dense particle. And this was found out by the person George Palade. Okay, so ribosomes is basically composed of what? RNA. So RNA is nothing but the ribonucleic acid. Plus, it will be having what? Proteins. Okay, so these are the structures which are mostly not surrounded by any membrane. So hence you can write it as not surrounded by any membrane. So two types are there of ribosomes if we consider the eukaryote as well as prokaryote. So if it's a eukaryote, then it's 80S, and if it's a prokaryote, then it's 70S. Okay, so now what is this S? S is nothing but sedimentation coefficient. Okay, so this both the things, that is 70S and 80S, they are having what? Subunits. Okay, so granular structures means what? If like it will be like small like this granules will be joined together. Okay, this is how the ribosomes look like. So mostly in and around the nucleus only you will find the ribosomes. Okay, so so basically what? The 70S and 80S, this is important. In eukaryote it's the 80S, whereas in the prokaryote it's the 70S. So just a second. Okay, so yeah. So as I was telling, 80S and 70S, these are the two subunits which are present. So in this, the 80S will be divided into 60S and 40. Whereas in the 70S, it will be 50 and 30. Okay, so S means nothing but the sedimentation coefficient. So sedimentation coefficient means whenever a cell, that is a prokaryote or eukaryote, when you centrifuge it, on what sedimentation rates does it settle down? On that basis only, it's the 80S and the 70S. So larger subunit as well as the smaller subunit. Okay, so mostly protein synthesis, that is the function of the ribosomes. Then comes the next one, which is the cytoskeleton. Cytoskeleton. So now what is the cytoskeleton? Cyto cell, skeleton. Okay, so this is nothing but the network of interconnected proteinaceous microtubules. Okay, so it's not necessary that only microtubules will be present. Sometimes it may be filaments. Okay, in the cytoplasm. So whenever there is a interconnected protein content, okay, of microtubules or the filaments in the cytoplasm, then that entire network is known as what? Cytoskeleton. So cytoskeleton, the main the function of the cytoskeleton is what? To maintain the shape of the cell, maintain the shape of cell, plus it provides what? Mechanical support. Mechanical support. And then the next organelle, which is the cilia and flagella. Cilia and flagella. Okay, so cilia and flagella, you already know what it is. So it's nothing but the hair-like outgrowths from a cell membrane. Right, so in this, as I told you, head, core, base, like that things are there. So here, the core which is there, it is known as axoneme. Axoneme. Okay, this is what the core of this hair-like structure. So axoneme is actually composed of what? Microtubules. And the arrangement will be 9 plus 2. So whatever the cilia or the flagella arrangement will be there around the cell, it will be what? 9 plus 2. That will be their arrangement. Hence, the core is called as what? Axoneme. So now what 9 plus 2 means, out of which this 9 which is there, it will be peripheral. So they will be what? Peripheral tubules. Okay, so these things are joined together by something which is called as radial spokes. Okay, so whatever the 9 peripheral tubules are there, they are what? Joined together by radial spokes. So if you have one this thing, what happens? Not, yeah. If you have one like this cilia or a flagella, so whatever the joining will be there, it will be present in double this one, or you can say it's a duplet. Okay, so this joining which is there like this is joined by whom? The radial spokes. Okay, so usually 9 is nothing but the peripheral tubules which we can see, and the 2 which is there, this will be the central ones. Okay, central microtubules which are bounded by a sheath, means the covering. Okay, so both cilia and flagella, they arise from what? The basal body, which is nothing but the cell membrane. Okay, so again, for this, there is one more structure from which it arises, which is called as centriole. Okay, so from the centriole-like structure, there will be emergence of this cilia as well as flagella. So cilia and flagella, again, as I told you, the core is called as the axoneme, okay, which are made up of what? Microtubules. And the arrangement basically of the cilia and flagella, that's 9 plus 2 arrangement, where the 9 is nothing but the peripheral tubules, whereas the 2 is the central tubule. Okay, and peripheral tubule, they are always present in 2. So hence they are joined together by something called as a radial spokes. Okay, then comes the next organelle, which is the centriole and centrosome. Very important. Centrosome and centrioles. So during the chromosomal division, mitosis, meiosis, this plays a very important role. Okay, so what is this centrosome or centriole? So centrosome, okay, they actually contain what? Like two perpendicular, cylindrical structures. Okay, and that two perpendicular cylindrical structure is only called as what? Centriole. Okay, so centrosome contains what? Two perpendicular arranged cylindrical structure. And this is only called as what? Centriole. So again over here, the centrioles are made up of 9 peripheral fibrils of tubulin protein. Okay, so centrioles are made up of what? 9 peripheral fibril, which is made up of what? Tubulin protein. Plus it has one central proteinaceous core, which is called as hub. Okay, so like how we saw previously over here, this thing was duplet, right? This peripheral tubules over here in the cilia and flagella. In the same way, here in the centrosome, okay, whatever the peripheral tubules which are 9, they are in triplets. Okay, so I'll just write it. The peripheral tubules are triplets. Okay, and each of this peripheral tubule will be connected to this hub through the same thing which we saw previously, that is the radial spokes. Okay, so the peripheral tubules, they are triplets. They are connected to the hub by what? The radial spokes. Okay, so this centrioles are only responsible to form a basal body for the cilia and the flagella. Plus, one more function it has is, okay, the functions if we see, first is the centriole form basal body of cilia and flagella. Plus, as I told you, it's very important during the mitotic and meiotic divisions. So at that time, what it will do is it forms spindle fiber during cell division in animals. Division in animals. Okay. Then the next organelle is the nucleus. Very important, brain of the cell, nucleus. Okay. So basically, this is a structure which is bound by what? An envelope called as nuclear envelope. Of double unit membrane. So two membranes it will be having. Okay, so this has majorly like 3 to 4 contents in it. Like first one, if you see, is the chromatin. Okay, so chromatin means nothing but it's basically a structure which is going to contain your nuclear material or a genetic material, which is primarily the DNA. Okay, with the DNA, it will have something called as histone proteins. Then non-histone proteins plus the RNA, or you can write and RNA. Because see, if it's a like virus or something like that, then the RNA will be genetic material. Other than that, the genetic material will be what? DNA. Okay, so this chromatin is usually during in the nucleus it's present, but most probably you will find a precise structure of it during the phase called as interphase. Okay, so there, like when you see the mitotic and meiotic divisions of a cell, there will be what certain phases like interphase, metaphase, like that, four, 5 phases will be there. So in that, whatever this nucleus is there, most prominently you can see during the interphase. Okay, so basically that nucleus is containing this thread-like structures in the initial form, which is nothing but the chromatin, which is consisting what? The genetic material. So this nucleus is surrounded by a nuclear envelope. Okay, so in between it is having certain fluid like things as well, which is called as what? Cytoplasm or nucleoplasm, you can say. Nucleoplasm. Okay, and there will be a space between the double unit membrane, and that space is called as what? Perinuclear space. Okay, plus one more thing it has is a nucleolus. So nucleolus also is found in the nucleoplasm, but it doesn't have a membrane. That's why it's called as what? Nucleolus. So if they say nucleus, means it will be having a double membrane plus nucleoplasm, okay, DNA and all those things. But if it's a nucleolus, it doesn't have a nuclear membrane. Okay, and where it is found? It's found in the nucleoplasm itself. But this particular nucleolus is an active site for rRNA synthesis. So rRNA means what? Ribosomal. Okay, so whenever there is a division, like whenever there is a mitotic or meiotic division, certain things happen in the structure of the chromatin. Okay, so initially what happens is, first the chromatin condenses and it is going to form chromosomes. So it will be like this. This will be what? Chromatin. And when it becomes chromosome, like this. Okay, this is what? Chromosome. So when the chromatin material condenses, it will become chromosome. So this happens during what? Cell division. And where does it take place? Nucleus. Okay, so if you see the structure of the chromosome, each of the chromosome will be having what? Two chromatids. And this is getting attached to something called a centromere. Okay, later on, whatever the centromere is there, that gets converted into a structure that is a disc-shaped structure called as kinetochore. Okay, so the chromatin condenses to form a chromosome during the cell division, and this chromosome is having what? Two chromatids on it, which is initially attached to the centromere. But then later, when the process is happening of the cell division, the disc, it converts into a disc-shaped structure, which is called as what? Kinetochore. Okay, so there are different positions of the chromosomes in the nucleus during the cell division. Okay, so based upon that, different types you're having. Okay, that is, if you see, if you take based on position of the centromere where it is present, then the first one we have is the metacentric. So metacentric means what? Middle centromere. Exactly in the middle, the centromere will be present. Okay, then you have the submetacentric. So here, little bit away from the middle. Okay, that is slightly away from the middle. Then you have the third one, acrocentric, which is near the end. And lastly is the telocentric, that is terminal. Okay, so these are what? The position, based upon the position of the centromere, types of centromeres you have. Okay, so there are like few chromosomes which also has a small structure within them, which is called as a satellite. Okay, not all the chromosomes possess this. Actually, this is called as a secondary constriction. So, which is satellite. Okay, at the constant locations, they will be having the satellite. But all the chromosome doesn't have this satellite. That is the main thing. Okay, so what are the functions of the nucleus? So because it's the brain, so obviously it will be having what? Certain functions, such as first thing is what does the brain do initially? It stores the information, and it also controls that thing. Okay, so it controls the cellular functions. Then it also controls what? Synthesis of proteins, such as controls the synthesis of structural proteins. Okay. Third one is it stores the information that is genetic. Why it stores the information for genetic is it will be helpful for the reproduction. During the reproduction process, it will be helpful. Okay, plus the behavior. For these two reasons, it is helpful. And because of this only, it also functions in one more thing, is in the induction of genetic variations. Okay, so these are all the functions that a nucleus performs. Then you have certain small bodies which are present in the cell organelles, which are called as microbodies. So microbodies is nothing but plenty of membrane bound, vesicles will be found in a cell. Those things are only called as microbodies. Okay, so membrane bound vesicles. And this membrane bound vesicles are having what? Enzymes. So this may be present in both plants as well as in animals. Like for example, say peroxisome and glyoxysome. So you will find the peroxisomes in plant cells and animal, whereas glyoxysomes in only plant cells.